My name is Qing Mao. I work as a bioinformatics analyst at Complete Genomics, located in San Jose, California. Today, I will be sharing with you one of the research projects I have worked on, named Advanced Whole Genome Sequencing and Analysis of Fetal Genomes from Amniotic Fluids. Amniocentesis is a common procedure conducted on pregnant women, usually for cases where a higher risk of pregnancy complication is suggested, or abnormal, non-invasive prenatal test is a result is found. The procedure involves insertion of a needle through the uterus and then collecting about 20 mL of amniotic fluids. And then these samples are centrifuged and separating the fluids, separating the cells from the fluids. The cells are then cultured and analyzed through either fluorescent in situ hybridization or in some cases microarray to look for abnormalities in chromosome copy numbers or large-scale structural rearrangements. In some cases, nucleotide changes are examined for a small number of specific genes. However, currently, thousands of genes have been described to be associated with genetic diseases. Therefore, the limited information we obtain from the amniocentesis is not sufficient to provide us for potential disease-causing genetic defects in the fetal genome. And it will be nice to be able to conduct whole genome sequencing with these samples to provide more information. At Complete Genomics, the standard sequencing procedure starts with a library construction, which involves the fragmentation of DNA and the insertion of adapters into these fragments. After library construction, the fragments are then amplified through a rolling circle process to form DNA nanoballs, or DNMs. Each of these DNMs contains hundreds of copies of the same DNA fragments, and these DNMs are then attached to nanoarrays and are subjected to sequencing by ligation. In addition to the standard procedure, we have also developed long fragment read libraries, or LFR libraries. These libraries, to prepare for these libraries, the long DNA fragments are first compartmentalized into 384 well plates. Then within each of these wells, unique barcode is attached to the DNA fragments. Then after sequencing, the barcode information can be used to phase the variants called in these genomes and providing haplotype information. Therefore, the first question we asked is, can we make high-quality DNA libraries with amniotic samples for whole genome sequencing? In order to sequence these fetal genomes from amniotic samples, the samples are first centrifuged to separate the supernatants from the cell pellets. From the supernatants, cell-free DNA was isolated and were sequenced using the standard library procedure. For the cell pellets, in addition to the standard libraries, we also attempted to make LFR libraries. For each of the families participating in this research project, we also obtained blood samples from both parents, and these samples were undergo standard library construction process and the sequencing process so that we would have genomic information for parental genomes as well. In total, we collected 33 amniotic samples from 30 families. And out of these 33 samples, 28 of the cell-free DNA generated successful standard genomes for sequencing, and all of the 33 cell samples provided successful standard genomes. In addition to that, we were able to make 12 successful FR libraries for sequencing. So in order to assess the overall quality of these fetal genomes, we looked at the coverage, the genomic coverage and exome coverage for each of these genomes. And we looked at the total number of variants called within each of these genomes. In these plots, each of the black dots represent a single individual fetal genome, and the average across samples for each library type is plotted as a dashed line. From these plots, we can see that up to 97% of the genome are confidently covered, meaning that up to 97% of the genomes, we were able to make confident calls for both alleles for all three different library types. And for the standard libraries generated from both cell-free DNA and the cell pellets, the exome coverage were above 98%. We also looked at the total number of variants and including SNPs, small indels, and substitutions. And we found about, on average, there are about 3.9 to 4 million small variants called in each of these genomes. This number is very close to total number of variants called in East Asian samples from 1000 Genome Project. Taken together, these data suggest that we were able to make high-quality sequencing library from both the cell-free DNA and the cell pellet of the amniotic samples. Since we were able to make more, for most of the cases, more than one library per sample, we were able to make comparisons of small variant calls between these replicates. The plot on the left shows the overlap of small variant calls between replica samples, and each of the blue dots represent the comparison for SNP calls, and the red dots are comparisons for all types of small variant calls. We can see from the plot on the left that the overlap between replicates is very high for small variant calls, over 99%, on average over 99% of the small variants called in one library can be also found in another replicate. Because we have the parental genome information, we were also able to count the overlaps between the parental and the fetal genomes. Similar to the results of from the left, we were observed over 99.5% for SNP concordance in this comparison, and also above 98% of the concordance for all small variant calls. Again, this result shows we were able to make reproducible genomes from different sources and different library types. Plink analysis is an effective way of showing sample relationships when plotting the Z0 versus Z1 from the IBD analysis. The expected location of pairwise comparisons is shown here in this plot. For example, for replicates, the data are expected to be located close to the 0 point. When we perform the plink analysis with all the small variants called in these fetal genomes and the parental genomes, all the data are located as expected in this plot, demonstrating the sample relationships from the genome data. Taken together, yes, we demonstrate the feasibility of making high-quality DNA libraries with amniotic samples for whole genome sequencing, and we were able to achieve high coverage genomic data. And all three types of genomic data have been shown to be highly reproducible. Therefore, with this data, we asked, can we identify de novo mutations in these fetal genomes? In order to discover de novo mutations, the variants called in the fetus genome are first compared against the parental genomes using complete genomics CODIF algorithm. This algorithm identifies variants that are present only in the fetus genome, but are not in the parental genomes. To filter out false positive variant calls in the fetus genome, it also uses variant quality scores and read coverage information in the fetal genome. And also to filter out false negative calls in the parental genomes, read coverage and confidence score of reference calls are used as well. The resulting variants are then filtered against population databases such as Personal Genome Project, 1000 Genome Project, Wilderly Project, and internal database we maintain at Complete Genomics to further remove potential false negative calls in the parental genomes. Through this process, on average, about 65 de novo mutations were found per genome for both standard library types, and on average 50 de novo mutations were found in the LFR libraries. It has been shown, it has been described that older fathers tend to contribute a higher number of de novo mutations than younger fathers. So we plotted the total number of de novo mutations in each of these fetal genomes against the age of parents at conception. In these plots, the color-coded dots, each of the color-coded dots represent a single genome. And from the plot on the left, we can see a positive correlation of total number of de novo mutations with the age of the fathers. And from the plot on the right, no such positive correlation was observed for maternal age. To get an estimate of the accuracy of our de novo mutation calls, we compared these calls between replicate samples. And from the plot on the left, we can see on average over 80% of these de novo mutation calls are shared between replicates, showing the reproducibility of our genome data. We also looked at base substitution spectrum of both inherited and de novo SNPs. For each library type, the spectrum for de novo SNPs is not significantly different from those of the inherited variants. So to get a more accurate assessment of the de novo mutation identification, we contacted and collected newborn DNA samples from 13 of the participating families and performed the Sanger sequencing using this DNA and also parental genome DNA. The first step was to see whether these predicted de novo mutations are present as heterozygous mutations in these children's genomes. And out of all the successful Sanger sequencing, over 90% of the predicted de novo mutations are indeed found as heterozygous mutations. For those that are called in more than one replicate, over 99% are confirmed, indicating that using replicate libraries is will improve our de novo mutation call capabilities. The next step was to confirm the de novo mutation status for these heterozygous mutations. And out of the 89 successful Sanger sequencing for trials, only 3 of them were found to be inherited, showing a low false negative rate of the parental genome variance cause. So this table is a list of all the coding de novo mutations we found across the 30 families. And we identified a de novo mutation in one of the genes named CHD8. This gene has been shown to be, mutations in this gene has been shown to be the causes for autism. Therefore, we contacted the family and confirmed that at the age of 2 years old, the child is showing macrocephaly, which is a common autism phenotype. However, at this point, the child has not been evaluated for ASD, but it has been recommended for close monitoring of development for this child. Therefore, we were able to make reproducible de novo mutation calls between the replica libraries, and we discovered a single fetus with a de novo mutation in CHD8 ASD-related gene. Beyond the de novo mutation calls, there are a number of analyses that can be performed to gain more information using this genomic data, such as copy number variants and structural variants calls, and also genes that are related to diseases or that are drug targets. Two of the families, families 12 and 21 and 22, were recommended for amniocentesis for a positive test of karyotyping for trisomy 21. And in this plot, using the read coverage data from all 3 library types, we can see a clear extra copy of chromosome 21 confirming the trisomy and we observed a similar pattern for family 22. It's very challenging for making accurate CNV and SV calls using short read sequencing data. Therefore, we attempted to make an estimate of the accuracy of our CNV and SV calls from the standard libraries. The plot on the left is showing the overlaps of CNV and SVs between the fetal genomes and parental genomes. And over 94% for CNVs and over 96% of SVs found in the fetal genomes are found to be overlapping with at least one of the parental genomes. When we looked at the overlap of the CNV and SVs to 1000 genome project, over 95% of the CNVs and 74% of the SVs are overlapping. Out of the potentially inherited CNV and SVs, we compared the overlapping rate between the replica samples, and the over 95% of the CNVs and over 85% of the SVs were found to be overlapping between replicates. All these data suggest the CNV and SV calls could be potentially accurate, and we can use this information to annotate. With annotation of this information, we might get more info. We might get more information for genetic defect that are harbored in these fetal genomes. These plots are showing the the distribution of PLI score and the missense z-scores for genes with rare variants, which are having minor allele frequency of less than or equal to 1% in population databases. The ExAC PLI score is and missense z-scores are constraint scores showing the intolerance of genes for loss of function mutations or missense mutations. When we looked at the distribution of the scores for amniotic samples compared with the agent samples from Personal Genome Project, the patterns are very similar. A majority of the genes are having low PLI scores or missense z-scores. It will be interesting to look at in detail the variants in genes with PLI scores of above 9.9, which are highly intolerant to loss of function mutations. And potentially these variants could be causing genetic defects or phenotypes in these newborns. Another analysis we performed was to look at the variants in drug target genes. This plot is showing number of coding variants in drug target genes, normalized by per million total variants found in each of the genomes. We looked at these numbers from amniotic samples, the parental genomes, and a number of different ethnic groups from 1000 Genome Project. The information gained from here could potentially be useful for drug prescription or medication for these individuals. So to summarize, we demonstrated the feasibility of creating high-quality whole genome sequencing data using amniotic samples from both the cell-free DNA collected from amniotic fluid or the fetal cell pellets. The genomic data we gained from both sample sources are highly reproducible, and de novo mutations were discovered by comparing fetal genomes with parental genome data, and we discovered a single fetus with a de novo mutation in an ASD-related gene, CHD8. With the availability of the whole genome sequencing data, a lot more analysis could be conducted, such as CNV and SV analysis, identification of the disease-related variants, and variants in drug target genes, and so on. This project has been a great team effort, and from sample collection to library preparation and data analysis. Here I would like to thank all the authors contributing to this project. And this project was supported by Complete Genomics, BGI, and Beijing University Shenzhen Hospital.